It gives me pleasure in welcoming all of you to guest lecture by Prof. Koudy from Institute for Regenerative Medicine, Wake Forest School of Medicine, North Carolina, United States of America. On behalf of Department of Chemistry, Faculty of Mathematics and Natural Sciences, IPB University, I would like to welcome Professor Koudy to our university, especially to our department, even by virtual meeting. We are glad to see you. I would like to express my gratitude for your time in giving lecture today. As your title of lecture, Tissue Bioengineering, is very interesting. I believe everyone here, me, my colleagues, and also my students would have an engaging, enriching session ahead. Thank you very much, Professor Koudy William. I hope Professor Koudy will continue having guest lecture for the following time. I even pray when the pandemic ends and everything runs normally, Professor Koudy Williams will personally visit our department and can contribute to guest lecture firsthand in person. Also, I would like to thank Dr. Irma Sparto as moderator and for arranging this general lecture. Okay, without further ado, let's join this guest lecture together. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Prof. Dia. So with this welcoming speech, that means we will be able to start the lecture, but you know, we always love to take pictures. Usually Koudy Williams is the photographer when he comes to Indonesia.
Oh.
Pictures. Yeah, but today we will take just a picture in front of this Zoom. So we have only a couple of panels. So please, Ibu Santi, you can start taking pictures maybe.
Yes, thank you, Bu Irma.
Open? Yeah.
Yes, on cam ya.
Maybe it's better if the slide should be removed first.
Professor Koudy, I will stop share screen. Can you stop share screen? Will you be able to start again?
Un, siap-siap ya Un.
Okay, we want to. Okay, we just make it small. How about it?
Okay, wait a minute. You want me to do what here?
Stop share screen. Is it possible, Koudy? You're afraid? Okay, don't worry. Don't worry. Don't do it. Oke, mungkin ambil aja ya.
Oke.
Langsung aja.
Langsung aja ya.
Yes, please.
Iya Bu, tapi ininya apa share screen-nya belum di stop.
Nggak usah.
Nggak apa-apa Un.
Nggak apa-apa.
Saya bantu ya.
Biar Uni aja Bu. Oke Un.
You only have 2 screens ya.
Ini di foto dulu ya Bu.
Oke. Oke. Sudah ya Yun? Oke. Sudah?
Foto slide pertama 1,
Ya.
2,
So second slide. Ya.
3. Sebentar. Sebentar ya.
While we're waiting, I would like to welcome also to Ibu Yesi. Ibu Yesi is from the physical.
Yeah, good morning Ibu Irma and Bu Dia Iswantini and also Professor William. Nice to meet you.
Oh, nice to meet you.
And thank you for the invitation and the opportunity.
Dr. Yesi is a very productive researcher also in working together with biomaterials. Okay. All done, Yuni?
Ya, sebentar Ibu belum. Slide yang kedua.
Kayaknya cukup, sudah ada.
Ya,
Sudah
oke deh.
saya.
Okay, thank you, everybody. Now it is such exciting. We are going to listen to Prof. Koudy Williams give a talk, but I would like to give a quick CV. He graduated in this, I have to say it, in 76 from Iowa State University and become a veterinarian in the 1983. And he works many places, and most of all in Wake Forest University, North Carolina, as a professor in regenerative medicine, comparative medicine, in pathology, in surgical sciences. That's where, and also Translational Science Institute. Many chapters, many book chapters and articles. And he has so many Indonesian students as his PhD students, and including me. Professor Koudy was also my advisor. So it's very great honor. Please, Koudy, Professor Koudy, you can start the lecture and share your perspective. Thank you so much.
Hey, thank you, Irma. Great to see you again.
Yes.
So I've come to Indonesia now about 21 times. I come about either every year or every other year to Indonesia. And I'm very sad I can't be there now, but hopefully things will clear up and I can be there within the next year. Irma has invited me to give many talks, mostly dealing with primatology, physiology, pathology, and that's basically what I am. She finally found something that I had to ask for slides for this. I probably shouldn't be telling you all this, but I got a C in chemistry when I was in school. I did a little bit better in physics, but I got a C in chemistry. So hopefully I won't come across too naive, but I'll go ahead and get started. Greetings from Wake Forest University. I work at the Institute for Regenerative Medicine, or as we call it, D.V.M. Hello. And it's not changing my...
You hit the slide and then you press enter or forward.
Well, that's what I'm gonna... There we go.
Yeah.
So D.V.M. history. We were established in 2004. We were the first regenerative medicine institute with primary appointed multidisciplinary faculty under one roof. We have more than 450 members, scientists from 56 countries. And if you'll notice down there right below the world, a little bit to the right, with a box around it is the Indonesian flag from our collaborations with Indonesia. We have 395 academic and clinical collaborations, 79 international. We were the first to demonstrate that complex layered tissue structures can, I'm gonna move my, there we go, can be engineered using cells, developed the first tissue engineered product to go to the US Food Drug Administration for a phase 1 approval for clinical applications, consisting of cells and biomaterials for injectable therapy. First to use biomaterials alone with and without the addition of cells. I still don't like where that is. There we go. First to create a laboratory grown organ that was successfully implanted in patients, and first to create functional solid organ experimentally. Identified and characterized a new source of stem cells derived from amniotic fluid in the placenta, which show promise for treatment of many diseases. Working on more than 30 different kinds of tissues and organ types. Selected to lead the nation research efforts, the Armed Forces Institute for Regenerative Medicine, embody on a chip, 14 ongoing planned clinical trials at D.V.M. currently. Our mission is to improve patients' lives through regenerative medicine. The vision is to be a leader in the global transformation from treatments to cures. A very big emphasis of ours to get this into people as safely and as quickly as we can. Our core values are innovation, teamwork, and integrity. Our mission is supported by 4 key objectives: to advance discovery through multidisciplinary structure, train and educate, which we all do, accelerate clinical translation, and to serve as a magnet for collaborations in regenerative medicine, which I just said we do, and including with Indonesia. We have within our institute multidisciplinary and cross-functional core programs. And regenerative medicine is a combination of basic, really a combination of basic sciences: molecular biology, cell biology, physiology, pharmacology, chemistry, nanotech, biomaterials, tissue analysis, bioreactors, bioprinting, and imaging. We can take our findings that we have in the lab and the benchtop through preclinical studies, then regulatory studies, then using a good manufacturing practice unit, and then into clinical translation. We have many different facets of what we do. Important is translation. We have IP and legal people that work with us, government affairs people that know how to work with the FDA, external affairs people, PR, finance administration, research administration, regulatory affairs, and education, all within 4 different levels in one building, 4 floors on one building, all of this. So which makes it very, very nice for collaboration, easy to go talk to somebody, easy to get things done. So what do we do? Regenerative medicine is more than growing replacement organs. It involves tissue engineering, cellular therapies, diagnostic platforms, healing therapies, and supporting technologies. My emphasis is really using small molecules and growth factor delivery into tissues that need regeneration to try to stimulate in situ regeneration. We have the ability to take our studies all the way through, let's see if I can get that, all the way up to phase 2 clinical trials. Then we will go start a new company and take it into phase 3 clinical trials and a product for surface. We have several standalone companies usually required for phase 3 studies, and this really accelerates our ability to translate our studies. 450 members, I'm in there somewhere, I'm not quite sure where. Anthony Atala is our director. We have 14, actually we have about 17 ongoing planned clinical trials right now. And our objective is to improve patient lives by developing regenerative medicine therapies and support technology. Okay, so today I'm going to talk to you about one aspect of regenerative medicine, and that's biomaterial science. I borrowed these slides with kind permission of Dr. David Williams, who is basically the founder of biomaterial science. He has written many, many books. He is the leading, he lives in South Africa, works with us, and is just a leader in this. He has written many books, including the Essential Biomaterial Science books. And he has really defined this new science, which began rather simply. And I think if you get anything out of this, I want you to get the idea of the challenge of trying to define what tissue engineering is and how intricate and advanced and how much further we need to go in order to bioengineer tissues. In general, we cannot regenerate those parts of our bodies that have fallen asunder. Whereas a newt or salamander can regrow limbs at will, our own pathetic attempts usually end with functionally useless scar. Now, of course, we'd rather have the brain of a human than a newt, and our evolutionary pathway determined that we were better off with good brains. But the question arises, if we're so smart, then why can't we engineer nature to equip ourselves now and then with regenerative power of a newt? Tissue engineering is defined as a creation of new tissue for the therapeutic reconstruction of the body by the deliberate and controlled stimulation. That's important. Deliberate and controlled stimulation of selected target cells through a systematic combination of molecular and mechanical signals. Another definition, a little bit more simple, is it's the use of cells and biomaterials and suitable molecular or physical factors alone or in combination to repair or replace tissue to improve clinical outcomes. So what's the challenge? Well, you have a disease, a defect or injury, you either need to replace the organ or tissues. With a loss of mechanical or physical function, might be able to replace it with a medical device. For a loss of certain organs, may be able to go to transplantation. When a loss of function is biological or physiological, devices are not usually applicable, and organs for transplants are scarce. For instance, for every 7 people that need a kidney transplant, there's only 1 available. So that means 6 people do not get it. We attempt to regenerate technologies such as tissue engineering. So as you can see, where and how are biomaterials relevant to these options? So that's what we're going to talk about. So this is a unmet clinical need by diabetic foot ulcers. Can this condition be treated by an implantable device or a drug? The opportunity for tissue engineering. What materials are required to engineer tissues to repair this horrific injury? Well, one of the things we have to do is we have to replace the nerves. And what we've done now is, if you look along the top, I don't know if you can see my cursor here, but if you look along the top there, there are several different conduits. You put 2 ends of a nerve in there, and then you create a nice environment for that nerve to regrow. You can do that by creating little honeycombs, little other different kinds of spaces for cells to grow. You can charge this with molecules that promote growth, put cells in there that promote growth, and then you apply physical or like electrical stimulation across it to cause those nerves to go together. So biomaterial, I just showed you one, is a substance that has been engineered to take the form which is used to direct by controlled interactions with components of living systems. For instance, that conduit is in the shape of a nerve. You put 2 ends of the nerve in and you stimulate it to go together. What are these procedures? What are the products? Well, implantable device for tissue replacement or assistance, temporary invasive devices, artificial organs or organ assist devices, drug delivery systems, injectable systems for enhancing imaging, non-viral vectors for gene delivery, and tissue engineering systems. That's a lot. I mean, that is really a lot you have to bring to bear on this problem. And it is getting more and more complex. The more we know, they always say the more you know, the less you know. And what we're finding is all these things must be brought to bear on these problems. So what kind of materials do we have? Well, many. Anywhere from plastics, I'll give you a little bit more detail on this, mechanical, ceramics, polyethylene glycol, there are chemicals, chitosan from udang, seaweed, ceramics, gold, silver, many different, and this types of materials, chemicals, and let me just show you. For instance, class 1, there are metallic systems made up of titanium. I don't know if any of you have had a joint replacement or had to have something fixed, but they put in titanium plates nowadays. Platinum, cobalt alloys, silver, gold. I'll explain to you how all these come together. The ceramic systems with alumina, calcium phosphates, bioglasses, iron oxides, rare earth compounds, quantum dots. Polymer systems, thermoplastics, elastomers, textiles, hydrogels, biopolymers. Carbon materials like graphite and carbon nanotubes. Composite materials that are some combination of the class 1 through 4. And engineered biological materials. And I'm going to talk to you right at the end, talk to you about pericardium and decellularized matrices that we're using now. Okay, so what should these materials have in them? If you're going to put them in the body and you want them to regenerate tissue, you want them to be functional, what guides us? Well, it's kind of impossible, but you have to have strength. Can't be too weak, but not a major factor. For instance, if you put in an artificial artery into the arterial side of the body, it must withstand all the stress forces and pressures that are on the left side of the body. Elasticity, that artery has to expand and contract. Optical properties, usually not relevant. Electrical properties, unless you're trying to stimulate growth with electricity, not that important. Magnetic properties, not usually relevant. Now, here are important ones. Biostability. The trick is, get a polymer, get a material. We want it to degrade eventually because we want it to be taken over by natural tissue. But it can't degrade too fast or it's gone away and the new tissue hasn't formed and it falls apart. Lack of toxicity, of course, you can't have that. Biocompatibility, the body cannot build a horrendous immune response to it and basically destroy it before it ever has a chance to form. But since tissue engineering involves the deliberate and controlled stimulation of selected target cells through a systematic combination of molecular and mechanical signals, the biomaterial should be able to deliver those molecular and mechanical signals. So it's not good enough just to put in a polymer and hope something good happens. Put in some cells on it and hope something good happens. Kind of where regenerative medicine started, but we've expanded and gotten much, much more sophisticated since then, and are now incorporating molecular and mechanical signals to aid in the regeneration of that tissue. Can't just put in the biomaterial and let it sit there. There has to be added things like cells and growth factors, and I'll talk a little bit more about that. Okay. For most implantable medical devices, the most appropriate host response is no response, right? You don't want it to attack it and wipe it out. The biomaterial should perform and continue to perform the required function without inducing any adverse response, usually achieved by maximal inertness. However, it's a problem. With tissue engineering applications, if there's no host response, there'll be no tissue regeneration. It'll just sit there like an inert, it's putting in something inert and just leaving it underneath the skin, nothing's going to happen to it. So the most appropriate response is involved with interactions that deliver molecular and mechanical signals. So you want it to interact with the body, but only in a good way. Again, you want it to interact, but only in a good way. This may be the most important failure mechanism in tissue engineering. Very, very important to failure is that you put in something that body attacks it, but it doesn't have a chance to release those signals that talk to the body basically so that you get regeneration. Okay. Let me move this a little bit. There are a few mechanistic insights associated with on what biocompatibility is. Classical views on biocompatibility have failed to predict significant effects attributed to implants. That's because the body has a very, very sophisticated system to attack foreign objects. Even worse, in preclinical studies, the immune system of a cow or sheep is very different than immune system of a primate, whether it be a non-human primate or a human being, and very different from a cat and a dog. And so if you're going to test something out, you got to test it out eventually in the species that you want it to work in. So you have to make those react with the species you're trying to put them in. There's little correlation between the in vitro data on biocompatibility. That's why that's just what I just said, is a lot of times in vitro data do not predict in vivo, and a lot of times in vivo preclinical data don't predict clinical studies. Many applications of biomaterials do not involve implanted devices, so classical wood healing may be irrelevant. Traditional views of biocompatibility do not allow for the idiosyncratic nature of human responses to external stimuli. Again, humans are very different. We're not newts, nor do they reflect the influence of the local and systemic conditions of the host. Again, immune system, very different. Humans also have enzymes that other animals do not have, vice versa. These views do not adequately reflect the fact that biocompatibility is not a property of a material. It is a property of a biomaterial host system. Hence, there is no such thing as a biocompatible material. You have to have a biocompatible pair. This is a very complicated slide. Don't worry about the details. I think I told Irma, yeah, you can go ahead and get PDFs of these if you want to, and people can go over them in more detail. But what this slide basically points out, there's blue, green, and sort of yellow, red down there at the bottom. What it is once you put a biomaterial in, several things must happen. You must have absorption of macromolecules. That is, it's going to be exposed to the body in its initial immune system. It must get used to that. It must survive that. It must also survive mechanical environment. For instance, if you don't have the right cues in there, a implant that you want to become a bone will become collagen, a joint, but it will not become bone. So you have to, it has to withstand that. And then the physiologic environment. There's going to be inflammation. There's going to be stresses, clinical procedures related stresses, and you must have the body react to that and begin to establish the cell-host interaction and biotalk that goes back and forth. Very important. Cells and matrix talk to each other. And if you don't have a biomaterial that can do that, it's not going to turn out the way you want it to turn out. Very important that all three of those are met by any biomaterial you put in of any size and any shape. In order to achieve spatio-temporal control, we need biomaterials that can be formed to a suitable template. Okay. One thing that I haven't said yet, and it's very, very important, is a biomaterial must create a niche for cells to grow in, for nerves to grow in, and it must be spatially correct for those cells to go in. You must make a nice home for them where they want to live, basically. And it's very important that they have that nice home, and that where they are beginning to produce the biomolecules that you want them to. biomaterials must be formed with a suitable template, which may be preformed, injectable, or printable, has the appropriate mechanical and biophysiologic properties, can incorporate and release necessary specific biomolecules, degrades with the appropriate kinetics, not too fast, not too slow, and lacks of toxicity. When those materials degrade, they can't produce toxic chemicals. It has the appropriate biocompatibility characteristics. But the biomaterial alone cannot perform all the required functions. It needs to be in the form that has appropriate architecture, forms a niche, and morphology, and needs to convey and deliver relevant biomolecules. A tissue engineering template comprises a biomaterial, a proper architecture, and biomolecules. Sometimes those biomolecules are produced by the cells you put in there. And very important, this template has to stimulate the cell niche. We'll talk about that. This slide basically just outlines this. So if you look at the kind of crescent-shaped blue there, that is basically the space that the tissue must form. It must interact with neural, metabolic, physical forces, paracrine forces, structural constraints, and humoral from outside, that body forming hormones, etc., to make that all work. And as you can see, that's a lot of things to bring together for a tissue to grow. So it's getting more and more complicated as we begin to understand more of what these cells and these biomaterials must have to be successful. This is what some of them look like. You can make these, you can make little nanoparticles, you can make, I think this is more silver and probably maybe gold and maybe some kind of, I'm not quite sure what else is in there, but can you see the different forms and the different little squares? It looks like a checkerboard. And you can make a biomaterial have these little compartments where cells can crawl in and matrix can crawl in, and it can form, and then you want that checkerboard to disappear someday. So it's complicated. Mandatory, the material should be capable of recapitulating, the architecture of the niche of a target cell. Moreover, since the cell niche is changeable over time, the material should be capable of adapting constantly changing microenvironment. For instance, if you put something in for the first 2 weeks, it's going to have a very different metabolism associated with that implant than it is later. Things are going to change. There's going to be changes in the amount of oxygen available to that implant, the growth factors that are being formed. As the cells begin to talk to each other, they're going to create a whole metabolic environment there, and the implant must be able to adapt to that. It should have elastic properties, particularly stiffness. For instance, you put in an artery or a heart valve, it has to be able to withstand the pressures of the heart. If you put in a bone implant, it must be able to hold and maintain the limb or wherever it's put. It should have optimal surface or interfacial energy characteristics to facilitate cell adhesion and function. What that means is the surface of that biomaterial has to be, for a better word, charged, so that it can accept and will attract cell adhesion. It can be done now with nanoparticles. Used to not be able to use those, but we can now. Very important that all that happens. Okay, specifications for template biomaterials. The material should be degradable if that's desired. Some you don't want them to go away. Most of them you do, with appropriate degradation kinetics, not too fast, not too slow. And I know I'm repeating myself, but these are important factors. Appropriate morphological and chemical degradation profiles. You don't want it to degrade and create something toxic that will chew up the tissue around it. The material should be injectable or printable if that's desired, with the appropriate rheologic characteristics and transformation mechanics and kinetics. Where necessary, the material should be compatible with the processing techniques that simultaneously pattern both material and living cells. What that means is you got to be able to successfully reproduce that design that you want from implant to implant to implant to implant. You can't vary a lot of those things and expect that implant to behave the same. It has to be consistent. It has to be the same if you want it to react the same. Okay, porous solids. This is kind of where we began. I think you probably heard about biospinning, where they used hydrogels, and I think I have a picture of it coming up. But the standard biodegradable synthetic polymers prepared by standard fabrication routes are unlikely to provide optimal tissue engineering templates, and prior FDA approval of the materials used in a medical device is not an appropriate specification or template. This is how we did electrospinning. It's like a spider web, and the polymer solution comes out of the syringe. It's jet initiated, high voltage power supply. It turns it into basically string. A collecting target, it can be round, it can be flat. And this is the what it looks like. Now that's nice. It has lots of holes and potential niche for cells to crawl in there. And you can make these polymers so they degrade slower or faster. They're usually not very strong if it's just a simple polymer solution, but I hope you can understand each one of those is going to look different. You can control the diameter a little bit, you can control how it's made. That the next one picture I could show you, it'll look different. And that means you have the potential of it not behaving similarly. This is kind of, here's where we've gotten with it. And this is really cool. This first one is basically you put a polymer on a plate, it spreads out and doesn't have much strength. It doesn't really have much form. So what they do now is they have a series of biomechanical, let's see if I can do that. Oopsie, sorry. What if I can do that now? No, I cannot. I wonder if I can. That's all right, I can wing it. What they put now to make something is they do a lot of inkjet printing, and they will put down various aspects of a tissue. They will put down structural, here in blue, which could be anywhere from gold, silver, something to give it form. It can be collagen, it can be fibrin. And then they also put down here in pink, they'll put down something that's functional. For instance, a growth factor, and they'll make it into a plate or a circle. And so what you want is you want that structure to support the functional aspects for a while until it goes away. Now they've gotten more sophisticated and they put in sacrificial structural things. So here's something you kind of want to keep around. Let me see. That can be gold, that can be silver, that can be something that just really doesn't go away very much. Then you have functional, and then they have what are called sacrificial biopolymers, and those are strong for a while, and they are put in there simply so they'll go away. They'll go away pretty quick and allow the function to occur, while the ones that the structural things they put in there to last a little bit longer stay there longer. So this is like a very complex sandwich that they put in, which contains functional, structural, sacrificial chemicals to make a implant. So as you can see, this is getting very, very, very sophisticated. So what's the process of all of this? You get something you like, it seems to be working. your biochemists, your chemistry people came up with something really good. So what do you have to do? Well, I have to do some proof of principle studies, then some chemical characterization. Always do literature reviews to make sure somebody didn't do it first. Confirmation of theoretical safety, may need independent verification. In vitro ex vivo assessment of cell function and tissue development in the template. Includes degradation, mechanical transduction, and matrix expression. Then you got to do some small animal studies, usually rodents, then large animal models for biocompatibility, functional testing, and one thing this did not include safety. First of all, safety. A lot of times I use sheep, dogs, or primate models doing good laboratory practices, and then you submit the data and documentation to the FDA for first in man experience. So, what are the definitions of materials for the 21st century? Well, regenerative medicine are therapies that treat disease, congenital conditions, or injury by the regeneration of functional tissue or organ structures. Functional, not just space taking. Responsive biomaterials are a biomaterial whose physical state is designed to have predictable responses to an external environmental conditions. They must be painstakingly put together. They must be consistent so that when you go from person to person to person or animal to animal, you're putting in the same thing. It's like you want to put in the same chemical to a reaction in the same concentration. It's the same principle there. It's just this is a whole gamish, a whole bunch of chemicals put together. A tissue-inducing biomaterial is a biomaterial designed to induce the regeneration of damaged or missing tissue or organs with the addition of cells and/or bioactive factors. Okay, organ on a chip. Cell culture models. This is really cool. This is an up-and-coming technology. They're cell cultures integrated into a microfluidic device to mimic biological activity, dynamic biophysical environments, and biochemical functions given tissue and organ system. These are three-dimensional, and they consist of a variety of cells, and they've actually been shown to predict reactions to drugs very, very well, which might be a very important part of the development process for any kind of technology. Bioink, cell-laden biomaterial that can be integrated into additive manufacturing processes to print 2D and 3D structures. Excuse me. Bacteriomylomics are the integration of computational tools, large databases, and experimental techniques to explore the basic material elements and combine them to discover and design new biomaterials and medical products. That would be, for instance, when I was talking to you about making those very complex multiple sandwiches of combinations of active, support, structure, all those different kind of chemicals that come together and to make a ideal implant that will stay around for a while, but will degrade over time and provide the important support it's supposed to. So I've been talking in generalities, but very, very important concepts there, and I hope you got that. So let's just talk about blood vessel engineering. What materials have they used? Natural polymers like collagen, elastin, hyaluronic acid, chitosan. The advantages are good cell attachment, good cell signaling, components of blood vessels, but they're mechanically weak and very expensive. Decellularized blood vessels, we've been doing that quite a bit. Good compatibility, mechanical properties of native vessels, difficult cell seedings. A lot of times once you take the cells out of it, it has to go through a decellularization product, and sometimes it does not create that niche that cells like, so they don't go in and set up home and grow. Poor cell migration due to the ECM structure. Synthetic polymers like PGA, PLA, PCL, cheap, ready available, tunable physical and chemical properties, important. You can make them kind of any size you want to. They can be toxic. Their mechanical properties for vascular tissue are not optimized yet. Suboptimal cell attachment and proliferation, at least so far, but they're getting better. Now, current constructs for tissue engineered heart valves. Seeded or pre-seeded natural biomaterials. Advantages are, you got the 3D architecture there. Disadvantages, immune response. If you put in a D-cell valve into a child, chances are that D-cell heart valve is going to have to be replaced 3-4 times. Cannot grow. It is going to slowly degrade, and is now being tested. Self or enhanced seeding natural biomaterials. Less time, uses nanotechnology and biomechanical to alter the characteristics. Unclear if mobilized cells form durable growing valves. Preclinical testing. We've actually done a little bit of work with that, where we've used D-cell valves and conjugate them with CD133, which attracts endothelial progenitor cells, and conjugate it onto the D-cell valve, put it in, we've used sheep, and the things will recellularize on its own. You don't have to add cells, you don't have to do anything. It will recellularize on its own, and that is under further testing right now. You can use seeded synthetic biomaterials, textile reinforced heart valves, and stent delivered heart valves. Now, that's really cool because many countries do not have the ability to do open heart surgery. A lot of people don't have the money, and the country don't have it. The ability, they don't have the, they don't have that many hospitals and that many trained open heart surgeons. So they've developed stent delivered tissue engineered heart valves. They go in through the groin and into the artery, and they can go up and plant them right in the aortic position or the AV valve position. And that is a lot cheaper, can be done in most hospitals. And so that is very good. Let me talk a little bit more about some of the biomaterials. Collagen is used quite a bit, actually quite a bit. Most abundant protein in the body, good cell interaction, tensile strength is consistent, but it is going to degrade. Fibrin, biodegradable, common natural product, but poor mechanical properties. It's very weak. Alginate, thickening agent for 3D bioprinting. It seems to be a good, but there's large batch-to-batch variation, expensive to produce, and so that kind of takes away some of the advantages. Hyaluronic acid, poor mechanical properties, cell-friendly using bioprinting. I have a bad right knee, and they have a new injection they can use to put right into your knee, which is basically hyaluronic acid, which forms, has your knee try to regenerate itself. Hopefully it'll work. Tyson, biocompatibility is okay, it's mechanically weak. PHA, shrinkage and valve regurgitation can be a problem. So as you can see, there are a variety of things they can use, nothing perfect yet. So if you go the route of getting a valve, or you get something and you want to get rid of the cells, and let's say you don't want to make a biomaterial, you just want to use the shape that's already there, you got to get rid of the cells. And there are various ways to get rid of the cells with series of detergents, freeze, thaw, osmotic solutions, which will blow up the cells or shrink them, detergents, enzymatic treatments, various things you can use. Nothing is perfect. Most of the time it leaves some residue and causes the existing collagen to cross-link, which reduces the ability of it to expand. Okay, recellularization chemicals. These are some of the chemicals they use. SDS, Triton, sodium deoxycholate, trypsin, ammonium hydroxide, or some combination of those. They all have their advantages. They all have their disadvantages. Nothing is perfect. Again, I think Irma can give you a copy of these slides so you can read them more carefully. Okay, whole organ decellularization. This is really cool. Instead of going with part of the organ, let's just decellularize the whole organ. And then try to, if it has bad cells or it has some damaged cells. So they've been doing this in the heart, the lung, the liver, the trachea, taking the cells out and then trying to put them back in. Here's one where they have taken all the cells out of a kidney. This is in a rat, and it's still very micro organized right now. You can't get real big arteries or big, organs to do this. You can see that you take away the cells, you leave the vasculature, which lets you then, once you get rid of the cells, maybe you can put bad cells, you can regrow that by putting cells back into the vasculature, and lo and behold, it seems to work. This is in renal cells, they decellularize the kidney and then put them back in, healthy cells back in, and we're able basically to form kidney structures by infusing normal cells into a decellularized kidney. That way you don't have to go and try to make a kidney structure. You don't have to worry about putting together complex biomaterial. You can get rid of the sick cells and put in healthy cells. There's some advantages to that. We've been doing liver studies where we slowly taking away the cells. You can see it turns red to basically translucent as we get rid of the cells, and then re-put in endothelial cells. And you can see over there in the right, the green fluorescence of putting in endothelial cells, which light up the vasculature and form vessels. Once you get the vessels in there, you have a chance of making that tissue healthy. Ooh, okay, I'm back, hopefully. Here we've done where we have native liver, put in a decellularized scaffold. You can see scaffold there, and then reinfuse cells, let it go for a long time, and you can see that scaffold is looking pretty much like a native liver, and it is acting like a normal liver. It produces hepatocytes, bile ducts, endothelial cells, and creates fairly normal albumin secretion and urea secretion, which is very typical of normal liver. So this is another option for a biomaterial. Just take the cells out of a sick, or even a cadaver, like a kidney or a liver, get rid of the cells that would cause an immune response and put in a person's own cells. Now, modifications of normal cells. So decellularized heart valve leaflets with recellularization potential. We've done that with the CD133. They've used magnetically guided recellularization of decellularized stented porcine pericardium-derived aortic valves. Modification of decellularized goat lung scaffold with chitosan and nanohydroxyapatite composite for bone engineering applications. So you can see what they're doing here is they are using decellularized tissue as their biomaterial and then trying to activate them to regrow. And they do that with nanoparticles. They can do that with a variety of other biomaterials like chitosan. They can use growth factors like VEGF. We've used CD133 antibody to do that. And other animal components. This is where I want to leave you. Traditional Chinese medicine has been using placenta medicinally for thousands of years. The first modern use of amniotic membrane was to treat skin wounds. in 1910, a long time ago. Anti-inflammatory, antimicrobial, antifibrotic, low immunogenicity, because it is from the placenta and hasn't seen a lot of immune cells. A lot of available, low cost, ease of use. So what they've done, what we've done here at D.V.M is dissection of an amniotic membrane, make a product out of it, basically freeze dry it and make it into a hydrogel or just an amnion. So that's your biomaterial. So it's amnion hydrogel or an amnion product, and used it in here. Okay, used it to try to heal skin lesions that we've created in pigs, and to look at the healing of these skin lesions. And these are just some pictures and images of what we have with the amniotic gel, the amnio graft, which is just the amniotic tissue, 3M patch, which is kind of standard that you put on wounds. And what you can see is the amniotic gel or the amniotic graft do a very good job of reducing contraction while forming epithelium, but reducing the size of the wound a lot better than a lot of the things that are on the market right now. Graft jackets, 3M patches do not do a very good job. So this is using a biomaterial that is from the amniotic membrane. So taking a natural amniotic membrane, and there's lots of those, and using that. So, conclusions. Well, it's a rapidly developing field of study, that's for sure. New techniques to aid recellularization of tissue products, cell-free implants, complicated, very sophisticated bioimplants made of a variety of biopolymers, metals, ceramics, and we're just beginning to realize that if we're going to do this, we have to produce a very consistent product that has the strength that it needs to, the biocompatibility that it needs to, be able to crosstalk with the cells in the body, degrade at a rate that you want it to degrade, get replaced with natural functional tissue. That is a tall order. That is a hard thing to do, but that's where we are right now. And that's what we're realizing we need to do. So with that, I will leave you. And certainly if you have any questions, I'll try to answer them, but remember, I got to see in chemistry. Thank you.
Thank you, Prof. Koudy Williams. Wow, that's a lot to take, but I'm so happy that the student already had some classes of some of the topics about your biomaterials and about concept of compatibility. So I bet they can interact. And I know you have several questions already on the chats. Would you like me to read, or would you like for them to directly ask questions? Please, what you prefer.
If you could get their questions and then ask me.
Okay, I will read it to you. So from Dr. Novian Darmawan, I have two questions. One, you mentioned in your talk the template of biomaterial is critical for biomaterial cell interaction. Is there any general guidance for designing the ideal template, such as porosity or large surface area?
Yeah, you know, if you look in the literature, people use a variety of porosities, use a variety of biomaterials. I just wanted to point out to you what they should do. And then you got to go searching for something that will do it. There are thousands of publications out there, people using various things. There's some of the nanoparticle stuff that's going on right now. They're making biomaterials that contain nanoparticles that then direct the cells. Very, very interesting. I'm not going to say one is better than the other. but porosity is an important issue. It has to be big enough to let the cells in, but not so big that they go in and then come right back out. You want them to adhere. You want them to be happy in there, and you want them to start producing the biomolecules they're supposed to. I don't have, what the ideal pore size or the ideal biomaterial is, for sure.
So it depends also on what you're going to design, right? So
absolutely.
Yeah, it's very case by case almost. Yeah.
Right. For a bone, you want it to get hard. Right?
Yeah. Yes.
I mean, you want it to get hard. Some other things you might want to keep elastic and with a lot of compliance, but a bone, you want it to basically calcify in and become a bone that can support weight. So it really depends on the tissue that you're trying to regenerate.
Okay. The second question from Dr. Novian also, is the decellularized method practical for real world use? How long does it take to get a full organ? Because I was thinking about that too. If you take it from a cadaver and then what do you think?
Yeah, I think, well, there's about 1,000 different decellularization techniques that people say is the right one to use. That's for sure. And it takes a long time. There's no doubt that it creates some change in the tissue that you're decellularizing. It's expensive. For instance, a cadaver heart valve that's been decellularized. They have these now that you can put in, well, pig valves but a human valve, if you get a decellularized human valve that's been decellularized the right way and according to the FDA and all that, can cost upwards of $20,000 just for the valve.
Or one valve.
One valve. And so it's very, very expensive to do that. That is why, and the other thing is it's hard if you're trying to put a valve in a child. Most of the valves out there that are decellularizing come from adults, right? You get old and you take it from somebody that's elderly and died, so the valve's way too big for a child. That's why they like the biomaterial that they can make, and they can hand make it in the size it's supposed to be. I think decellularized tissues, I sort of get the feeling that we've seen the best of them and they might be going away, but not until we get better with our biomaterials that we're trying to make. I think it's not going to last that much longer, but we have to come up with better biomaterials.
Okay. Thank you, Prof. Koudy. Pak Ian, is that enough?
Thank you, Professor Koudy. Thank you.
Thank you. So, well, I was thinking also the same question as Dr. Novian, because I cannot imagine you have to wait and it's more cheaper to just make one, right?
Well, it's certainly cheaper to do it, but again, you also are, the other problem is if you make it, there's a lot of countries out there that do not have advanced biomaterial science and able to make something like this. The trick will be to make them and have them been popular enough that the cost will go down. Kind of like those computers we used to buy that, well, the handheld computers that could add, subtract, multiply, and divide, maybe do a cosine and do some square roots for you. It used to be $300 and now they give them away. So hopefully the more you use them, the better they are. The price will go down over time. Like everything, when and your color TVs, the plasma screen TVs used to be $7,000, $8,000 a piece, and they're 100, 200, 300 now. It'll get cheaper, but not for a while. There'll still be a room for decellularized tissues. I like the idea of trying to get some sick tissue to repair itself in vivo. In other words, we're doing some studies with people with a pulmonary disease. They have a lot of fibrosis, and we're trying to come up with things we can put in to get that lung to repair itself in situ. You don't have to put in anything. You're trying to reprogram that lung to repair itself. Same thing with heart valves. You're trying to, that's what you're trying to do. And I think that's cheaper because a lot of these molecules aren't that expensive. For instance, we use, as you know, CXCL12 quite a bit, and it's not that expensive to use this stuff. And I'm kind of hoping that will tide us over until the biomaterials get up to speed of what they need to be.
Thank you, Prof. Koudy. We have 4 questions waiting for you. So from Hari Noviardi, thank you for the nice presentation. I have a question. What are the challenges in developing biomaterials from tissue engineering, and how long can these biomaterials be used as implant based on biostability and biocompatibility data? It's a general question. It's very important.
Where we are now is, and I think David Williams would agree with me, is I think we finally know what we need to have a biomaterial do. And I think that's very important that, we finally know what we need to know. And that's the beginning. I think we finally realize we got a long way to go, but that's the first part of true knowledge is you realize that where you need to go. And that's why I wanted to do this presentation, to show basically what it is we need to know to have a biomaterial that acts that way. Where are we? We're working on it as fast as we can, but like anything that you produce, you're still years away from using these widespread clinically, but it depends what it is. We already have some biomaterials we're using in clinical studies right now. They're more simple right now. They're a lot simpler. They're decellularized bladders, penile implant, vaginal tissue. As you know, we did a study in cats for kidney disease, and that is in clinical study right now. But the actual biomaterials, I think people are finally getting on board with what needs to be done, and that will take a while.
Yeah, it takes time. So yeah.
Yes, it does.
Also, lots of financial, financial aid.
Well, startup is always expensive. And as you know, I've come to Indonesia and tried to encourage some work done with using biomolecules and using that to help regenerate tissues. And the biomaterials will come along later, but as you said, there's a lot of startup money to get that done. The inkjet printers and all that are expensive, and it's not impossible, but they're expensive.
Yes. Okay. Thank you. From Adi Sifriadi, Professor Koudy, could you please explain about material characteristic for tissue engineering application, especially based on molecular interaction with the template? Molecular interaction.
Yes, very, very important.
Yeah.
Very important. That material you put in, let's say you start with a ceramic or you start with something like that, pretty soon you're going to want that to start interacting with the body. And that's why I showed that one slide, the complicated slide. It's got to know where it is, then it's got to start responding to the pressures that it's under, and then it has to start creating that cell-to-cell interaction and crosstalk that needs to happen. That is where the small molecules come in. It is where some of the nanoparticle stuff comes in. And there's even stuff where they've created a biomaterial just by the way that it's made, starts the cells in there and they start cross-talking. They don't add anything to it. It's a very simple design, and they put it there and the cells start talking to each other. But for heart valve, it's very important that the lumen cells talk to the cells in the inside the leaflets for them to work. If they don't, it'll just scar in and you end up with a scar. So they must talk to each other. Very important, very important. And it's that, therefore, you have to have a really good idea molecularly of what you're trying to accomplish.
Thank you. One interesting question about what is the prospect of to change tissue of hair skin? You know, we have problems with our hair. So what do you think about that? It's from Agus Winarso.
Okay, this is to grow hair?
Is that what you mean, Pak Agus? Okay, probably, Koudy.
Oh, okay. Well, first of all, you're gonna have to get in line behind me. And, but yes, you know, we have a study ongoing right now. Dr. Atala has a study going on right now with bioengineered, basically hair plugs that they're putting in right now. That, in fact, I'm on the Animal Care and Use Committee. We just approved it for so they can go ahead. And this is, God, what animal model? You know, there's the stump-tail macaque gets normal male pattern baldness, but this is a nude, some kind of nude mouse or rat that they're going to do this and put in basically bioengineered hair follicles and try to get those hair follicles to go.
Oh, that's a good news. Okay, so
as I say, they're going to have to get in line behind me.
Okay, so next is from Dr. Budi Riza. What are important criteria need to be passed when you want to use an artificial tissue made of biomaterial from animal tests to human body tests?
So what's the process of going from animals to people?
That's right. It's the criteria. What is the most important?
Well, to go to people, first and foremost, it can do no harm. That's number one. It must be safe. That's you must have a biomaterial that is safe and doesn't cause more harm than it helps. In fact, it should not cause harm. So that's a big one. And most of your last steps in getting approval to use it is a safety study. A lot of times those are done in monkeys because they have the immune system and the physiology that's most like that of human beings. It must be biomechanical, that is not causing too much of immune response. It cannot cause, if there's cells in there, they cannot turn cancerous on you. And so it increases your risk of cancer. They cannot cause a new disease, right? You treat one and then you end up with something else. It can't do that. So to get it to clinics, you got to do a series of safety studies. It must be efficacious. In other words, it must work. Got to work, but mostly you got to do a series of safety studies to make sure it's not causing any harm.
Thank you. Prof. Koudy, please be patient. I think we have 4 questions.
That's fine.
I will no more, or maybe we have that for one. But could you please stop share screen so we can see your everybody in one template?
Okay, do what?
Stop share screen. Stop share.
Stop share.
Yeah, because we have a black hole there. Nah, that's right.
There we go.
Okay, so the next question is from your favorite person, Silmi Maria. How to confirm the biomaterial is not rejected by the body before transplanted? Should be done with a pilot study or just trust the manufacturer that produced it?
Never trust the manufacturer.
That's right.
Never trust the manufacturer. No, you got to do the studies yourself, Silmi. You've got to do, is there any peripheral problems with rejection? In other words, you can do some peripheral white blood cell evaluations to see if you are stimulating various types of macrophages or T cells. You got to look around. A lot of people just look at their implant and say, oh, everything looks fine. You got to look around there where you put the implant too. You got to see what the regional lymph nodes are doing, what the spleen is doing. You've got to do your own. And I know you can do those there, but it's very important also to look for activated white blood cells in various forms, whether they be macrophages, T cells, lymphocytes, whatever. Yeah, you got to do your own.
So we
They may
can
make
do
you do
in
more.
vitro
They may
assay
make you do
before.
more.
Yeah. Mhm.
Yeah, but you got to
Okay.
do your own.
Okay, thank you, Prof. Koudy.
Thank you. Okay, so, okay, I hope you're, I can see your window is starting to get darker.
It is?
Evening, but you stay up, okay? Don't go to sleep.
I'm not going to sleep.
Okay, now it's from Rifan Nurfala. Hi, Professor. Would like to ask about properties of biomaterial and specification for template of biomaterial. How do we determine the right level of elasticity? And second, how to prepare the biomaterial according to the rheological characteristic? Oh, that's
yeah, that's why you always have, in any study design, you have normal controls, because the normal controls, and remember what I always used to tell you, your most important group in any study is your control group. And so you have to know what the normal is. Let's say you're looking at a bioengineered artery. Well, you got to get that artery, normal arteries out, and you got to do some biomechanical testing on it to know what the stress strain curves look like. And you've got to do that, and then test that against your implant. And your implant should try to do its best to mimic or recapitulate those biomechanical properties, the strength and the elasticity. So you got to do some controls. A lot of that is actually in the literature. If you go searching for it, a lot of times people have already done doing biomechanical tests is not new. And so a lot of times you can find that stuff. You still got to do a control study because you're going to have your own ways of measuring biomechanical properties, and it has to be against your own controls. But it'll give you an idea where you can start, right? If it has to have a certain strain modulus, then it will give you a place to start, so you don't have to go searching all over the place. Look at the literature, control group.
Yes. I always remember that before you start study, read a lot of journals and have a control. Yeah. Okay. Yeah. Now, one question from Dr. Nofian about the injectable dermal filler for aesthetic purpose, right? Especially for the face, because it's a trend now. What do you think the long-term effect after external exposure like UV and so on like that? Do you think there will be problems of those injectable dermal filler?
I don't think we'll really know, Irma, until these people have had those dermal fillers for years, to what happens to them. And, you know, people are getting them at a younger and younger age. I don't know why they needed that when they're young. But there are people that are doing it. We won't really know. Short term, there doesn't seem to be that much problem, but we won't know. We're 10, 20 years out.
Well, actually, we don't need it, right? Growing old, elegant.
No. I deserve every wrinkle I have.
Yes. Okay. We will have one last question, but it's in Indonesian, but I will try to translate from Ramdan Hidayat. So you mentioned about perturbation physiological environment, right? So
yes.
We program re-expression of the gene, right? And then what do you think about this? According to aesthetic, also this is about question of DNA salmon. Have you heard of that? There's a trend about DNA salmon that it can from extraction DNA from sperm of salmon, and then they inject it. Have you heard of that?
So DNA from salmon?
Yes, from the sperm. They said is to improve the skin also. So it's really a big business for this cosmetic.
Really?
So you should be one making innovation such as.
I can get salmon sperm and make millions of dollars.
Yes. What have you heard of that, Prof. Koudy?
I've only read a little bit about it, Irma. I'm not very conversant on it. I know people are injecting all kinds of stuff into them now, trying to stay young. I don't know about salmon sperm that much.
Okay. Pak Ramdan, maaf, I'm sorry. We need to read more about that. Okay. One more. What is it? Thank you so much, Pak Koudy. It's from everybody.
Oh, well, you're very welcome.
So many things in our head, and we always, you know us, we always want to learn and learn and learn, especially from such a person like you. But we do not want to keep you up. So it has been such a great talk, and I think this is a really good strategy to have you in the last session of our semester, right, Pak Novian? Yeah, because so the student already had some open perspective about the biomaterial.
Right. That's true, Irma. Yes.
Yeah, that's right. You kind of sum it up.
And I'm also suggesting students to continue the study and research for the cosmetic purposes, because it's very big business.
Again, you're going to have to get in front of the line of me.
But
I mean, yes, I mean, here in Indonesia, a lot of like Korean product cosmetic.
Right.
That they really forward, how to say, advance in the cosmetic for the application of biomaterial for the cosmetic.
Very progressive.
Right.
Progressive, yeah.
Well, I know it's big business and stuff. Just but it's like giving a drug. You got to know exactly what you're giving. You got to know the dose that you're giving it, or you're going to get different results. And it's got to be a consistent product, or you're going to get different response. Some people get great responses, some people won't. It's like taking platelet-rich plasma, or you get some cells. Big in veterinary medicine now is just getting some cells from animals and shooting it in their knees. It'll work sometimes, it won't work others, because it depends what kind of cells. And unless you know it and you've standardized it, you're going to get varying results.
Well,
I did check with David Williams. If you want to make PDF out of those slides, you're welcome.
I will, and I will share to this the code here. That was a very good closing statement, that we have to make sure about safety. So
yes.
That I will not make it add another statement, but most important, we would like to say thank you so much for
you're very welcome.
Sharing. And it looks like that you will be more, we will chase you more, asking of your...
Well, maybe sometime I can come over there and stay a while and like teach a semester or something.
Oh yes, that will be great. And then see what's really going on in real life, right?
Yeah, exactly.
Thank you so much, Prof.
Oh, you're very welcome, Irma. Great to see you.
Thank you so much for the audience, for the head of department, Pak Novian, and also all the lecturers that attended this lecture. It has been very insightful, and we are so happy. And we cannot give you a soto ayam or sate ayam, but at least you can dream about it. So
I can, and we had fruit tonight for dinner, and I put in mango. So.
Oh, that's nice. At least you have a touch of Indonesia there, right?
A touch of Indonesia. We can get the they're kind of yellow, called champagne mangoes, and we can get those here. And so I had mango tonight anyway. No durian.
Okay,
no
next
durian.
time we'll have it's a rain check. You will treat again.
Okay.
Thank you so much, Prof. Koudy.
You're very welcome, Irma.
Semuanya. With this, we say Alhamdulillah. Alhamdulillah, is praise to God that we finished all this. And so we hope to see you again soon. And may the pandemic get over soon.
I hope so.
And we get together again. Thank you.
I hope so.
Everybody. Thank you. Have a nice day, and goodbye. With this, I will close the lecture. Thank you.
Okay. Well, thank you very much.
Waalaikumsalam.
Thank you. Bye-bye.
Bye-bye.
Bye, Prof. Koudy. Thank you.
Hi, Silmi.
Kak
Good to
Silmi,
see
thank
ya.
you.
Terima kasih Bu
Ya.
Irma. Terima
Ya,
kasih.
sama-sama.
Prof Dia, makasih.
Bu Dia.
Say hello to
Irma.
Robin.
Oh, I will. I will.
Ya.
Bye-bye.
Well, thank you all.
Hope I can
Oh.
visit you again.
Terima kasih semuanya.
Makasih Bu Irma.
Ya. Ya, Assalamualaikum.
Salam.